Okay, so today we're going to continue our discussion of proteins, but we're going to delve in depth into enzymes and discuss enzyme activity, regulation, and inhibition. So we'll talk about first the enzyme-substrate interaction, a couple models of how those two interact, and then we'll talk about cooperativity. We'll discuss various mechanisms that enzymes use in order to catalyze a reaction. We'll talk about how environmental conditions such as pH and temperature impact the activity of the enzyme. We'll talk about kinetics of enzyme activity and specifically Michaelis-Menten kinetics. We'll discuss the 4 types of inhibition that you need to know for the MCAT, and we'll look at Lineweaver-Burke plots as well for each of those. And we'll end with talking about some other mechanisms of regulating enzyme activity, so positive and negative feedback, as well as a few enzymatic forms of regulation. So, just to start, the substrate is the compound that the enzyme acts on. And there are 2 different models that are used to explain how the enzyme and substrate specifically interact. So neither one of these models is completely true. It's probably a little bit of a combination of both. So the active site model, well, first we'll talk about what is the active site. The active site is the region of an enzyme where a substrate binds. And you're going to see me abbreviate enzyme-substrate complex as E-S complex. And that's also the site where catalysis occurs. So the active site model says that the 3D shape at the active site and the chemical reactions present at that site enable the specificity between the enzyme and the substrate. So more specifically, the active site model says that enzymes and their substrates interact in a lock and key fashion. So the enzyme, which is the lock, has a very specific shape and chemical environment that only one substrate, which is the key, is going to fit into. And I just wanted to add here, so you guys know that enzymes are often stereospecific. To be stereospecific, they'd have to be chiral themselves, and that means that they would only react with one stereoisomer of a particular reactant. So the active site model says the active site is in this fixed 3D orientation, and that is specific to one substrate. On the other hand, the induced fit model says that once a substrate binds to the enzyme, that induces the enzyme to shift its conformation slightly. And that shift in conformation makes the enzyme even more complementary to its substrate. So essentially, the binding of the enzyme to substrate increases the affinity of the enzyme for its substrate. So as the name proposes, induced fit. The binding induces an even greater fit between the enzyme and the substrate. So this one looks at the active site as a little bit more dynamic than the active site model, which proposes it's this fixed orientation, whereas the induced fit says there's a slight change in conformation once the substrate binds, and that is the best fit between the enzyme and substrate. So here are a couple of images that help depict these 2 models. So this is the lock and key, also known as active site model. So you see the enzyme has this specific shape of its active site, and that doesn't change, going from enzyme plus substrate to enzyme-substrate complex. On the other hand, the induced fit model, you see that the active site here undergoes a little bit of a conformation change. So it says the enzyme active site forms a complementary shape to the substrate after binding. So once this substrate touches down on the enzyme, there's a slight conformation change, and that is when the perfect fit is induced. And this is just depicting in general the interaction between the enzyme and substrate. Specifically, this is the induced fit model. And I will say the induced fit model is probably more indicative of what's going on. So the active site is slightly complementary to the substrate, but once they bind, the enzyme changes shape slightly, and it's a perfect fit at that point. Then the substrate undergoes a reaction and turns into products. So you have enzyme-substrate complex undergoing a reaction and turning into the enzyme-product complex. And then the products are released from the active site. Cooperativity explains how changes in the concentration of the substrate might affect the enzyme's activity. So enzymes have cooperativity, and other biological molecules do as well. So cooperativity specifically refers to proteins that combine more than one of the substrate molecule. So keep that in mind, because cooperativity says after one substrate binds to the protein either becomes less or more cooperative to binding to future or more of that substrate. So it would have to have multiple binding sites for one protein, on one protein. So positive cooperativity, this occurs when the binding of one of the substrates to the protein increases the protein's affinity for that substrate, making it easier for more substrates to bind to the protein. So an example of positive cooperativity, which we'll go into immense detail in a subsequent lecture, is hemoglobin. So hemoglobin is made up of 4 polypeptide subunits, and each subunit can bind to one oxygen molecule. And so one hemoglobin protein can bind 4 total oxygen molecules. And the hemoglobin protein exhibits positive cooperativity. So after the first O2 molecule binds to one of the subunits of hemoglobin, the other 3 binding sites shift their conformation slightly, making it easier for the second, third, and fourth oxygen molecules to bind. Negative cooperativity is just the opposite of positive cooperativity. So once one substrate molecule is bound to the protein, the other binding sites undergo a slight conformation change, which results in decreased affinity for subsequent ligands. So we know that in general, enzymes are going to increase the rate of a reaction by lowering the activation energy. And specifically, they lower the energy of the transition state. In other words, they make it easier for the reactants to reach that high energy transition state. But specifically, how do they do that? Well, there are a number of different ways that enzymes perform this function. We'll discuss some of the most common here. So, one of the ways that enzymes do this is they may preferentially bind to the transition state and stabilize it in that way. And what I mean by preferentially bind is that the enzyme interacts more favorably with the transition state than it does with the reactants or the products. And so that preferential interaction for the transition state that the enzyme has results in a stabilization of the transition state, because there's probably a number of intermolecular forces which stabilize the transition state. Acid-base catalysis occurs when a residue of the enzyme is actually going to participate in the reaction it catalyzes by donating or accepting hydrogens from the substrate. And we know that enzymes must be regenerated into their original form after the reaction. So if one of the residues donates a proton, it has to receive that protein back at the end of the reaction that it catalyzes, because it has to come out unchanged. Next, we have proximity and orientation, which refers to when an enzyme positions the substrates near each other, so that's proximity, and in the correct orientation for them to react. So it really just aligns them, aligns the two or there may be more than two, but it aligns the reactant molecules in the perfect orientation to facilitate that reaction. Metal ion catalysis is when an enzyme uses metal ions to assist in catalysis. So often those ions, which are cations, are going to stabilize the transition state in some way or another. And then there's covalent catalysis, which is when a residue of the enzyme participates in the reaction by forming temporary covalent bonds with the substrate. So just like for acid-base catalysis, the enzyme has to be regenerated into its original form after the reaction is finished. You should know that the enzyme may do any combination of these mechanisms, so they're not mutually exclusive. So here, this yellow arch here is indicating that the rest of the protein's over here. So this is just a hydroxyl group of one of the residues of the enzyme. And you see that this is an example of covalent catalysis. So you see there's a covalent bond that forms between the substrate and the enzyme. And then water is added, and then the enzyme hydroxyl group is regenerated. But you see that it's enabled the reaction in which this X group is substituted out for a hydroxyl group. So it's directly participating in the reaction through bonds, but at the end, the enzyme looks just as it did at the beginning. So here I've included 3 different examples of an enzyme catalyzing a reaction. And the way that it's depicted here is how you might see it depicted in a passage on the MCAT. So the residues are drawn out, the residues of the enzyme that are going to assist in the reaction specifically. And then the substrate molecule is also shown. And you see as the reaction proceeds, you can see that the enzyme residues participate in the reaction, and it may be through covalent bonding as it is here, or through metal ion stabilization, or acid-base catalysis, or any combination of these mechanisms. So this top row here shows an enzyme, actually a protease, assisting in the catalysis of a peptide bond. So specifically, this shows a cysteine residue helping the hydrolysis of a peptide bond, and here you see a serine residue. So in both cases, the cysteine or the serine, you see that residue engages in a nucleophilic attack on the carbonyl carbon of one of the residues that's going to be cleaved. So the substrate itself is also a polypeptide. So you see that there's a covalent bond formed between the sulfur of the cysteine residue and the carbonyl carbon. And then the cleavage reaction proceeds. So it's a hydrolysis reaction, it requires water. And you see now that peptide bond has been cleaved. And at the end, the cysteine residue is regenerated so that all the residues look the same as they did at the beginning of the reaction. And here serine does the same thing, engaging in nucleophilic attack and having this temporary covalent bond, which facilitates the reaction of the hydrolysis of the peptide bond. And then here's an example of metal ion catalysis. So the cation here is going to stabilize this tetrahedral intermediate in which we have a negatively charged oxygen. So there are a number of helper molecules or ions that enzymes often require in order to perform their biological function. In general, those molecules are referred to as cofactors. So cofactors are any non-protein molecule or ion that's going to help the ion perform its function. So cofactors is the broad umbrella term that all of these 4 fall under. And cofactors can include metal ions, coenzymes, vitamins, prosthetic groups, and really any molecule that you see as crucial to the enzyme's biological function. So metal ions are commonly cofactors. They're required for metal ion catalysis. So here we see metal ion catalysis occurring. This metal ion is not a part of the polypeptide chain of the enzyme. Instead, it's associated with the enzyme, and that may be temporarily associated or more permanently associated. But it's required for that enzyme's function, because without that metal ion, the enzyme couldn't perform metal ion catalysis. So some common metal ions that we see acting as cofactors are these I've listed here. Next we have coenzymes, which are non-protein, organic, so organic means not metal ions, helper molecules that are going to be loosely associated with the enzyme. That's also another pretty broad category. And then prosthetic groups are non-protein, organic helper molecules that are permanently or covalently bound to the enzyme. So prosthetic groups are like an arm and a leg of the enzyme. They're permanently bound to the enzyme So it's not a loose or temporary association, but it's a part of that enzyme. So the enzyme is characterized in part by the prosthetic group that is associated with it. So an example of a prosthetic group is the heme prosthetic group in hemoglobin. So this here is the heme group. And so here is this prosthetic group. This is an organic molecule. And in the center of the prosthetic group is a metal ion. And this is permanently bound to each subunit of hemoglobin, and it's required for the function of hemoglobin. So heme is the prosthetic group, and in the prosthetic group we have a metal ion. And then finally, vitamins, specifically those that are water-soluble, not fat-soluble. Many of those act as cofactors. So that molecule may be a precursor to a cofactor, or it may be a cofactor itself. So water-soluble vitamins that act as cofactors are generally vitamins B and C. So there are many different B vitamins, 6, 12, 3, etc. And an example of this is FAD and FMN, which we'll talk a lot more about in a later lecture, are electron carriers, and they're made from vitamin B2. Another term is holoenzyme, and that just means the enzyme, the complete enzyme, the whole enzyme. That's how I remembered it. So it's the enzyme complete with all of its required prosthetic groups, cofactors, whatever it needs to perform its function. And then, just so that you're familiar with this term if it comes up, apoenzyme is the sort of precursor. It's the enzyme lacking its cofactor or, molecule that it requires in order to perform its function. So apoenzyme cannot perform its biological function yet. It's not a fully formed or, completely functional enzyme. Holoenzyme is the whole enzyme associated with any molecules that it requires. Just one more thing to add about the terms on this slide. A vitamin, if you're unfamiliar with the specific definition of a vitamin, it's any organic compound that your body requires to function and can't produce in sufficient quantities itself. So essentially, it's an organic compound that we need from our diet because we can't produce it ourselves. And then the fat-soluble vitamins, so those that aren't water-soluble but are fat-soluble, are vitamins A, D, E, and K. So I remembered that as ADEC, A, D, E, and K. And you should know those as the fat-soluble vitamins. So if you know those, then all the rest are going to be water-soluble, of course. So a number of environmental conditions, such as temperature and pH, are going to impact the activity of an enzyme. So in the last lecture, we did touch on this. So we talked about how changing the temperature, changing the pH, changing the salinity of the solution, etc., is going to likely denature your protein if it's done to an extreme extent. So these changes in environmental conditions may denature a protein if they are dramatic enough, but they may also just not denature the protein, but just impact its function, either decreasing or increasing its rate of catalysis or render it unable to catalyze a certain reaction. So specifically, when we're talking about temperature, in general, increasing temperature at first, so when it's not too dramatic, is usually going to increase the activity of the enzyme. So usually we know that increasing temperature often increases the rate of reactions in general, right? Because it increases the energy of those reactant molecules, making it easier for them to get to that activated transition state. So generally, increasing temperature is going to increase enzyme activity, but that only continues to a certain point at which the temperature is so high that it begins to denature the protein. And at that point, of course, the enzyme cannot perform its proper function. So if you look at the last lecture, we had a chart. On the y-axis was enzyme activity, and on the x-axis was temperature. And as temperature increased, enzyme activity gradually did increase up until a certain temperature, which was the temperature of denaturation, and then the enzyme activity plummeted. So all enzymes have a specific optimal pH range in which they function best. And that pH range may be physiological pH around 7.4, but it may also be slightly acidic or basic, depending on where that enzyme is meant to function. So for example, digestive enzymes, many of them, which function in the GI tract, function most optimally at low pH because we have acidic conditions in our GI tract. Changes in pH from this optimal range are going to change the protonation state of the protein's residues and thereby disrupt its biological function, because if you change the protonation state, you may disrupt salt bridges, dipole-dipole interactions, and you may also disrupt residues which are required for the catalytic functioning. So remember that in the active site, you have some residues, like these guys here, and here, you have some residues that are absolutely required for the functioning of that enzyme. So not only does changing the protonation state of residues change the structure, but if it changes any of the residues which are required for the catalytic function, so those that might participate in the reaction, for example, then you render that enzyme completely ineffective. Whether or not the shape changes, if you affect the residues within the active site of the enzyme, then you're undoubtedly going to negate its biological function. And then, as I said, severe pH changes may denature a protein. So even if they're not severe and don't denature the protein, those slight pH changes, if they, change the residues in the active site or just shift the structure slightly of the enzyme, it may be either less functioning or not able to function at all. So now let's talk about enzyme kinetics. We know that in general, enzymes are going to increase the rate of a reaction, and they do so by lowering the activation energy and stabilizing the transition state. So those are one and the same. Stabilizing the transition state, meaning lowering the energy of the transition state, is the same as lowering the activation energy. So that means that enzymes impact a kinetic parameter and not a thermodynamic one. We went in detail about kinetic versus thermodynamic parameters in one of the chemistry lectures, but this is important to know. So enzymes don't affect thermodynamics of a reaction, meaning they're not going to change the delta G of the reaction. So they can't make a non-spontaneous reaction occur. That's going to require coupling the reaction to ATP hydrolysis. What they can do is increase the rate of a reaction which is already spontaneous. So if the reaction's already spontaneous, but it just has a really slow rate, then it may appear to not be occurring at all. And so an enzyme is going to increase the rate and make that reaction able to occur. You can have an enzyme working on a non-spontaneous reaction as long as I said here, you couple that non-spontaneous reaction to ATP hydrolysis or some other extremely spontaneous reaction. We'll talk about reaction coupling in a few lectures. So Michaelis-Menten is a facet of enzyme kinetics, which is really important for you to know for the MCAT. So Michaelis-Menten are two scientists who came up with the Michaelis-Menten constant and a number of other equations that attempt to describe the kinetics of enzyme catalysis. So if you have an enzyme plus a substrate, you know that forms the enzyme-substrate complex, and then the reaction occurs here, and then you have enzyme plus product at the end. We have a number of different rate constants, so the forward rate constant going from enzyme plus substrate to ES complex is referred to as K1, and then K negative 1 is going from ES complex to E plus S. And then finally this K2, that's also referred to as Kcat, K catalytic, because this is the rate constant for the actual reaction occurring, the catalytic rate of the reaction. Of course, increasing the concentration of the enzyme or the substrate is going to increase the reaction rate, because it increases formation of the ES complex, right? So if the forward rate, so the formation of the EX complex rate is equal to K1, concentration E, concentration S, then we're going to be able to increase the rate of forming that ES complex. Now the Michaelis-Menten constant, there's some algebra behind its formation, but just know it's a constant for a specific enzyme, and it's equal to K2, also known as Kcat, plus K negative 1 divided by K1. We'll talk a lot about that on the next slide. I'm just introducing some terms now. Vmax, V for velocity, is the maximum rate of catalysis when the enzyme solution is already saturated with substrate. So essentially what that means is substrate is in excess. So it's only going to depend on on the rate of catalysis, so K2 or Kcat, and how much enzyme is present, because when the enzyme is saturated with substrate, that means that all the enzymes are filled up with substrate. So adding more substrate isn't going to increase Vmax. But adding more enzyme could increase Vmax. So Vmax is equal to K2 times concentration enzyme. And it's also important to know that if we refer back to our discussion of rate laws, this is the rate determining step here, going from ES to E plus product, because this is where the actual reaction occurs within the substrates. So this is the rate determining step. That means the maximum rate of this entire thing is just the rate of the rate determining step. So that's K2 times concentration enzyme. And this is when the enzyme solution is already saturated with substrate. So this diagram here kind of shows at this concentration of the substrate, the substrate's in excess. So what really matters is how much enzyme is present and the catalytic rate. But how much substrate is present isn't going to affect Vmax at that point. And then, of course, this does depend on concentration enzyme, because if you add more enzymes, more reactions can occur. So more of the substrates can now enter the enzyme active site. And then this other velocity term, V0, refers to the initial reaction rate. And Michaelis-Menten came up with this equation here, and there's a lot of algebra behind this, but you don't need to know it. But V0, so the velocity at any time point, dependent on a certain concentration of the substrate, is equal to Vmax times concentration substrate over Km plus concentration substrate. So remember, this was Km, it's a constant for that enzyme. And so you need to know this here. Another thing to know about Km is the Km is indicative of the binding affinity for that enzyme. And understanding why it's indicative of binding affinity is going to be really helpful in the MCAT. So that is this reasoning here. The Km value is equal to the concentration of substrate that's required to have a velocity equal to half the Vmax. So what I mean by that is when the concentration of the substrate is equal to the value of Km, the Michaelis-Menten constant for that enzyme, then plugging that in here, so Km and concentration S are going to be the same value, what you get is V0 is equal to Vmax, let's substitute in just 1, times 1 over 1 plus 1. So you see you get V0 is equal to 1/2 Vmax. So I'll say that again. When the concentration substrate is equal to that value of the Km, then the velocity is half the Vmax. So that means if the Km value is higher, then it requires more substrate to get to half of the maximum velocity. And that indicates low affinity, right? But if you have a lower Km, then that means that less concentration substrate is required to get to that 1/2 Vmax. So that indicates a higher affinity. And that's how you can understand why Km is indicative of the binding affinity for that enzyme. As I said, higher Km, lower affinity, lower Km, higher affinity. The efficiency of an enzyme is a term that's equal to kcat divided by Km. So a high kcat would mean an efficient enzyme, right? Because it's churning out many of those enzyme plus products. So, that's high kcat or K2. And you want a low Km, because low Km indicates high affinity. So if you have a higher kcat and a lower Km, you have higher efficiency of the enzyme. So efficiency relies on both the catalytic rate and the binding affinity. So here is a graph of substrate concentration versus reaction rate. So you can think of this as the V0, and the V0 varies as a function of substrate concentration. So when this curve begins to level out, it's essentially going to reach an asymptotic point where it's approaching Vmax. So Vmax is indicated here on both of these graphs. And 1/2 Vmax is just 1/2 the value of Vmax, right? So this is around 0.32, so it's going to be around 0.16, around that value. It's just going to be halfway down on the y-axis. And we know that when the concentration substrate is equal to the Km, that's when we're at 1/2 Vmax. So we're at 1/2 Vmax at this concentration of the substrate, meaning that that's also the value of the Km. So that's how you can use this graph to find the Km. You first have to find the value of the Vmax, then find 1/2 Vmax, and the substrate concentration at which you're at 1/2 Vmax is also the value of the Km. Here this shows that the Vmax is reached when the enzyme is completely saturated with its substrate. And remember, this is specific for a certain concentration of an enzyme. That's because Vmax, of course, is dependent on the concentration of the enzyme. So this Vmax is going to vary depending on how much enzyme you have present. But it won't vary depending on the substrate because you reach a saturating level of the substrate. So this plot is great to use to find Km, but we can also take the double inverse of this graph, and what we get is something called the Lineweaver-Burke plot. So the double inverse, meaning we're graphing 1 over reaction rate versus 1 over concentration substrate, and that's actually going to give us a linear plot. So this specifically is used to determine how inhibitors impact the Vmax and the Km value of enzymes. So you're going to be very familiar with this. You see here, if you were doing an actual experiment and plotting the Lineweaver-Burke plot, you'd only have dots in this quadrant of the graph, right? Because you're not going to have a substrate concentration that's negative. And on this plot, there are some important points that you need to know. So first, the slope is equal to Km over Vmax. That's important to know. It's also important to know that the y-intercept, found here, is equal to 1 over Vmax, and the x-intercept is equal to negative 1 over Km. So that means using this plot, you can solve for Km and Vmax. So there are 4 different types of enzyme inhibitors, and they differ in their effect on binding and or the catalysis. So basically they may affect the affinity of the enzyme for its substrate or not, and they may affect the catalytic rate of the enzyme or not. And that means that they differ in how they change or don't change Vmax and Km. So the first of those 4 types is competitive inhibition. Competitive inhibition occurs when the inhibitor actually competes with the substrate to bind at the enzyme's active site. That means the inhibitor is going to mimic the structure of the actual substrate because it's also going to bind to the active site. And that means it binds to the enzyme, not the ES complex, which is different for the different inhibitors. Competitive inhibition can be overcome if you increase the concentration of the substrate. Essentially, because the enzyme and the substrate are both trying to bind to the enzyme. Sorry, if the inhibitor and the substrate are both trying to bind to the enzyme. If you increase the concentration of the substrate, you can sort of outcompete the inhibitor and make the effect of the inhibitor negligible, because you just make so much substrate available to the enzyme that the inhibitor has minimal effect. So that means that at a very high concentration of the substrate, the inhibitor doesn't affect the enzyme's rate, meaning there's no change in Vmax, but an increase in Km, because you're decreasing the affinity of the enzyme for its substrate. Well, you're decreasing the apparent affinity of the enzyme for its substrate because it interferes with the binding mechanism of the enzyme to the substrate. So it's important to know that while I say increase in Km, it's an apparent increase in Km. But the Km of the enzyme is sort of a characteristic of that enzyme, a property of that enzyme, and isn't actually changing. It's just that there's an apparent increase in Km because there's an apparent decrease in the affinity of the enzyme for the substrate when the inhibitor is around. So you see the inhibitor is going to mimic the structure of the substrate, binding at the active site and competing with the substrate, which is why it's called a competitive inhibitor. So here you see how it affects both of the plots. So a competitive inhibitor is not going to change Vmax, which you can see on this plot here. And on this plot here, we know that right here, this y-intercept is 1 over Vmax. So with the inhibitor present, the blue line, there's no change in the Vmax. But since the x-intercept is negative 1 over Km, because this is a larger fraction with the inhibitor, that means it's an increase in the Km. So for example, if this point was negative 1/2, and this point was negative 1/10, that means the Km here would be 2, and the Km here would be 10. So an increase in Km indicates a decreased binding affinity of the enzyme for its substrate. So the next type of inhibition is uncompetitive inhibition. And this occurs when the inhibitor binds to the ES complex, not the enzyme alone. And it reduces the enzyme's catalytic activity. So it reduces the ability of the enzyme to catalyze the reaction. It does not bind to the active site, meaning it binds at an allosteric site. So allosteric site is a site other than the active site. And because it binds to the enzyme substrate complex, meaning it requires the substrate to be present in order to inhibit the enzyme, it's a better inhibitor when the substrate concentration is higher. So that's important to note, because it requires the substrate to be there in order to do its inhibition. So if you have this kind of reaction going from ES plus inhibitor to the ESI, enzyme substrate inhibitor complex, as the inhibitor binds, forming more of these ESI complexes, Le Chatelier's principle is going to drive the formation of more ES complexes, because we have E plus S going to ES plus I. So by Le Chatelier's principle, the result of the inhibitor, of an uncompetitive inhibitor, is there's an apparent decrease in Km, or it looks like the inhibitor is actually helping the binding of the enzyme to its substrate. So though that's counterintuitive, the way that I sort of remembered that was that the uncompetitive inhibitor, it doesn't really actually do this, but this is how I remembered it. It sort of locks the substrate into its enzyme even though it prevents the reaction occurring, it sort of cinches them in. So it appears to decrease the Km of the enzyme. And of course it's going to decrease the Vmax because it impacts the catalytic activity of the enzyme. It decreases the maximum rate that the enzymes can catalyze the reaction. So this plot here shows that there is a decrease in Vmax. 2 is with the inhibitor. And you can see that the shape of the curve shows you why there's an apparent decrease in the Km. So this is then the Lineweaver-Burke plot. And the uncompetitive inhibitor is above it. It decreases the Vmax and decreases the Km. And the way that I remember the shape of the uncompetitive inhibitor is that it's uncompetitive. If you think of these as two racers running alongside each other, they're sort of uncompetitive. They're doing their own thing. They don't really cross paths. So that might not work for you as a mnemonic, but that's how I remembered it. And then also, it's called uncompetitive because it doesn't compete with the substrate for binding at the active site. So now let's talk about a mixed inhibitor. A mixed inhibitor is sort of a mix between competitive and uncompetitive inhibition. It's going to bind at an allosteric site of the enzyme or the enzyme substrate complex. So it's a mix between the two because remember, competitive binds at the enzyme and uncompetitive binds at the ES complex. It does decrease the maximum reaction rate, so it affects the catalytic activity of that enzyme. But its effect on Km is going to vary depending on whether it has higher affinity for E or higher affinity for the ES complex. So if that enzyme, if that inhibitor, a mixed inhibitor, if it acts more like a competitive inhibitor, meaning it binds better to the enzyme alone than it does to the ES complex, then it's going to result in an apparent increase in Km, just like competitive inhibitors do. If that mixed inhibitor acts more like an uncompetitive inhibitor, meaning it binds better to the ES complex than it does to enzyme alone, then, like an uncompetitive inhibitor, it's going to result in an apparent decrease in Km. So it does decrease the Vmax, so the Lineweaver-Burke plot is going to vary depending on its affinity for E versus ES complex. So it didn't include a Lineweaver-Burke plot. But know that the inhibitor, the graph of the rate plus inhibitor and without inhibitor, they're going to have the same y-intercept, but the line of the inhibitor may have a lower or higher Km. So it could cross so this is no inhibitor present. It could cross like that, or it could cross like this with a shallower slope. It would be like. So basically it could have a increased or decreased slope compared to the no inhibitor line, but they're going to intercept at the same y-intercept. And the last type of inhibition is non-competitive inhibition. So this occurs when the inhibitor binds at an allosteric site and decreases the activity of the enzyme. It does that by changing the conformation of the active site, making it less catalytically effective. It doesn't impact the binding of the substrate to the enzyme in any way. And it can bind to the enzyme or the enzyme substrate complex. So it just affects the catalytic activity of the enzyme, meaning there's no apparent change in Km when you add the inhibitor, but there is a decrease in Vmax. So here you see the substrate binding site or the active site is different from where the inhibitor binds. And the inhibitor combined with or without the substrate present. So the inhibitor and substrate bind independently of each other. Either or both may be bound at any time. And then completing the reaction depends on the enzyme undergoing a conformational change that's blocked by the inhibitor. So what that means is the inhibitor basically affects the ability of the active site to catalyze the reaction. So here you see a non-competitive inhibitor is going to decrease the Vmax. And then when you look at this plot, with the inhibitor present, you see that there's no change in Km, but a higher y-intercept. So this would be like 10 versus 2. Higher y-intercept is going to mean decreased Vmax because this is 1 over the V. This is just a combination of the plots for the types of inhibition. Non-competitive is not the same as mixed, so you can kind of cross that out, but this was a good combination plot minus this error here. So you should know what the effect of each type of inhibitor is and whether it binds at the allosteric site or the active site, and whether it binds to enzyme alone or enzyme plus substrate complex. And here's another summary chart of the different types of inhibition for the normal plot. So we have a couple more things to talk about. One of those is feedback regulation. So this is one of the ways that enzyme activity is regulated. So feedback regulation is when the product or a later made compound of a reaction pathway feeds backwards and allosterically regulates an earlier enzyme of the pathway. And the name kind of implies it is a feedback regulation because this later product goes back to an earlier step of the pathway and either increases or decreases the rate of that step. So of course, if the product goes back and decreases the rate of the enzyme, then overall you're going to have less product made out of that pathway. So there's negative feedback and positive feedback regulation. Negative feedback regulation occurs when a product or a later made intermediate of a pathway inhibits an earlier step in the pathway. And negative feedback regulation is the more common type. And that makes sense because if there's already a lot of the product being made and it's starting to accumulate, then the pathway needs to slow down in order to conserve energy and resources. So if the pathway is churning out a lot of the product and it's starting to accumulate, then the product is going to start to feed backwards and inhibit an earlier step, meaning it's going to decrease the amount of the product being made because there's too much of it. Positive feedback regulation is less common but occasionally occurs, and that's when a product or a later intermediate of the pathway is going to increase the activity of an earlier enzyme, overall increasing the amount of product being made. So this is a negative feedback loop and this is a positive feedback loop. You can read this on your own, but this is basically what I just said. It blocks an enzyme in the beginning of the pathway, and that occurs when there's a buildup or excess of the product being made. And this occurs in order to achieve homeostasis in the cells, to prevent this pathway from occurring in overdrive. So you see the product literally feeds back and inhibits an earlier enzyme. And feedback inhibition could also be this later intermediate going back and inhibiting this earlier enzyme or this one. So I wanted to show this diagram here, which is a little more complicated way of looking at negative and positive feedback, but it's the type of diagram that you definitely see on the MCAT. So you see the pathway here is PIP2 going to PIP3, and then that's going to activate AKT, resulting in production of either this or this. So here's the positive feedback of the pathway. So the AKT activation results in phosphorylating this, which activates this, and then this guy is going to downregulate the amount of this. And if you downregulate the amount of this enzyme, then that means less of the reverse pathways occurring. In other words, more of the forward pathways occurring. So that's a sort of indirect form of positive feedback, because decreasing the backwards enzyme is going to result in less of the reverse reaction and more of the forward reaction. And then this is negative feedback. So this product, so the phosphorylation of this guy here activates this, which activates this, and that's going to inactivate this forward enzyme. So less of the forward rate occurring. So positive feedback may not be a direct activation of an earlier enzyme. It could be inactivating the enzyme which is acting in reverse. So it's a sort of indirect form of positive feedback. A few more terms for you. Allosteric enzymes are just any enzymes that are going to increase or decrease their activity when some compound binds at their allosteric site. That compound may be referred to as an allosteric regulator. And the allosteric site, as I've said, is just any site on the enzyme other than the active site. So the binding of that allosteric regulator causes the enzyme to shift its conformation slightly, and that shift in conformation is either going to increase or decrease the affinity of the enzyme for its substrate or the catalytic rate of the enzyme. Next, we have covalent modification of enzymes. So these are all other forms of regulating the activity of enzymes. So covalent modification occurs when a group of atoms is covalently added to or removed from an enzyme, changing the activity of that enzyme. So usually another enzyme is required in order to catalyze this addition or removal of the group. The most common type of covalent modification to enzymes is phosphorylation or dephosphorylation of enzymes. Kinases are the other enzyme that do the phosphorylation, and phosphatases do the dephosphorylation. This enables really tight control over enzyme activity. It's like a switch turning the enzymes on and off. Usually phosphorylating an enzyme turns it on, and dephosphorylating it turns it off, but it could be the reverse. So there are other forms other than phosphorylation and dephosphorylation, but this is the most common type. And the covalently linked group, which for this is a phosphate group, may either increase or decrease the enzyme's activity. Finally, a zymogen. This is a term that refers to the inactive form of an enzyme. And that inactive form has to be modified in some way in order to perform its biological function. So the zymogen is modified, and that turns into the fully functional enzyme. So zymogen is just enzyme precursor that doesn't have its biological function. Usually, the zymogen is cleaved in some way in order to convert it into the active form of the enzyme. So the purpose of this is mainly to prevent enzymes from doing damage by performing their function at the wrong place and the wrong time. So if you produce an enzyme in its active form always, then if that enzyme is around its substrate, it's going to start catalyzing the reaction. And you don't always want that. Sometimes you just want enzymes in certain regions to be active. So a great example of this is the digestive enzyme pepsinogen. That's the zymogen. So often zymogens, the name is just the name of the enzyme, and then you add ogen. So pepsinogen is converted into pepsin when the zymogen is exposed to a low pH environment. So the low pH environment in the GI tract is crucial to activating this enzyme, because you don't want pepsin going around and performing its function when it's not in the digestive tract, because that would be really destructive. So here's an example of phosphorylation. We have our protein and then our phosphorylated protein. So you see the addition of the phosphate group has changed the conformation of the protein. That's what it's depicting here. And the phosphorylation is done by a kinase, which uses ATP in order to, as the source of the phosphate. And then the dephosphorylation is done by a phosphatase. So here's an example of digestive enzymes, and it's sort of a chain of activation from zymogen to active form. So often the transition from a zymogen to the active enzyme requires another enzyme. So you see enteropeptidase is the enzyme here that turns trypsinogen, the zymogen, into trypsin. Then trypsin acts to convert all 4 of these zymogens into their active form. Okay, so that ends our discussion of enzymes for this lecture.